This is a special episode of the Immunology Podcast, Immunology 2023, Day 1. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. As you all know, today launched the start of the Immunology 2023 conference, an annual meeting of the American Association of Immunologists here in Washington, D.C., and Brenda and myself and the Immunology Podcast team are all attending. Drop by the Immunology Podcast booth in the exhibit hall to play some games, win a prize, and find out how you could be featured on an episode of the podcast. Today and every day throughout the meeting, we'll be releasing special episodes discussing our favorite sessions of the previous day. So if you weren't able to attend, we've got you covered. We're going to kick off things in just a moment. Before we get to that,
are you studying natural killer cells? If so, make sure to join us at STEMCELL Technologies workshop presented at Immunology 2023. During this workshop, Dr. Amanda Durkin and Peter Morin will be covering tools to optimize your natural killer cell research and presenting on cutting-edge NK cell isolation and expansion products to help stay ahead in this rapidly evolving field. The workshop will take place on Saturday, May 13th at 11:15 a.m. in the Exhibitor Hall Workshop Room 1. For more information on STEMCELL Technologies workshop and other activities, make sure to visit STEMCELL Technologies booth at number 4011 or learn more at stemcell.com/AAI. And at the AAI meeting, we are. Jason, are you excited?
I am, and we got a little bit of taste today. We had one session from 2 to 4. Everyone's settling in. You know, the hotel check-in lines are long. The coffee lines are moderate. I think everyone's gearing up for a great conference.
Yeah, for sure. So, I mean, a lot of people, a lot of familiar faces around. Very exciting. The place is huge, and there's so many talks. It was so hard to choose which session to attend, I have to be honest.
Well, so I just go with my phenotype because I think it's easier. And I went to the metabolic and microbiota effects on autoimmunity. What'd you end up at?
Well, I went to one of the sessions on B cells and helper T cells. So it's like B cells, but they're not alone. The T cells are always there.
Well, we've established that you're a B cell, right?
I am. So I guess you know it's a kin situation. So yeah, this B cell and T helper cell responses during viral infections. That was the session, and very interesting. Let me give you some highlights of the talks. There were I think it was 8 talks. First, I really liked there was first talk by Chris Risley from the Francis Lund lab. So we had her on the show. So it was a trainee from her lab. And it was talking about multipotent memory T cells. And so, you know, on cue with the Lund lab research. And I thought it was very interesting because we were looking at the role of TBET on the development of memory cells and how the expression of TBET allows or relates with the ability of memory T cells to reactivate into effector B cells upon like a secondary stimulation. Very interesting. Basically, they show that TBET marks this particular subset of memory B cells that are quicker to become effector memory B cells, and is required. So the expression of TBET is required for this rapid differentiation. And then not only that, but it also seems to improve the maintenance of memory B cell populations in general. So very cool presentation by Chris Risley. And then we had also Ariane Schippers from the Victoria Lab, and also a very cool, in this case, they were looking into antigenic imprinting. And I think this is so relevant because they're from Rockefeller University, by the way. So they were looking at the effect of an initial antigenic exposure into a secondary. So how, if you have already one exposure, one vaccination, for example, how does this affect de novo, like new B cell clones developing on a secondary exposure? And then if you have a third exposure, then where is this memory B cells coming from, the first or the second line? And I think they made a really good case showing that that the first response dominates over the second response if you have the same antigen. And I wanted to just highlight the model that they used, this mouse model in which they had a fate mapping mouse model in which they had a flag tag attached to, I think it was the IgG kappa chain, and then they could use a Cree-inducible system with tamoxifen to then change this initial flag tag into a strep tag. And therefore, they could differentiate antibodies that were generated throughout from the first response. And then if they had they infected the mice, they allowed memory to generate, they gave tamoxifen, and then they infected the mice again. And so then you have, after the second infection resided, then you had two memory populations that you could differentiate by either strep tag or flag tag, and you knew that this flag tag came from the second exposure and the strep tag from the first. I thought it was very cool. Basically, they look into this idea of the original antigen sin, or I think antigenic sin, showing that the primary response really dominates. There were a couple of was another talk also on that from Tanushree Dangi from the Peñalosa lab at Northwestern University. And again, they also were looking into pre-existing immunity and how it modulates further responses in the case of mRNA vaccines. And again, they see that in their case, they kind of conclude that by having a pre-existing response, and a pre-existing antibodies, that you limiting, because if you have a second infection, then because it gets cleared so quickly, you limit antigen expression, and that impairs B cell responses following an mRNA vaccination. And this seems to be, this acceleration of antigenic clearance is dependent on humoral, so of course humoral immunity, and through FC dependent mechanism. They did some experiments in which they had the FAB part and the FC and the FC part separate. So a lot of like how subsequent responses are affected by previous ones, I think is very interesting. It will help us understand better vaccination. And I don't know, do you want to talk about some talks from your session?
Sure, I can jump in here. So this had a mix. It's autoimmunity, but then metabolic and microbiota effects on it. So
okay.
So I'll start with the metabolic, and then you can jump back in. So a bunch of talks trying to break this off. When one talk was about microRNA 21 and B cell autoreactivity. It was from Kristen Bricker. I could not get all the PI's names in here. We had 15 talks in the 2-hour session.
It's just 8. Come on.
And more for mine.
Really?
Yeah.
And it was 10-minute talks.
They had less than 10 minutes. And,
really?
And they barely 10, and they went crazy. And I couldn't get. They passed the title slide faster than I could record everything, and that's not in the app. So I have the authors' names and the abstract names, but I couldn't get everything. Some furious typing here. All right, so they have a TLR7 duplication autoimmune spontaneous mouse model, and they show that this loss of this mRNA 21 leads to reduced germinal center responses and reduced CBRL expression with less cytokines. And so they show that this is indeed a CBRL dependent system. So long story short, in the first paper, TLR7 dominated autoimmunity, which exists in people as well, is driven in part through MIR21, and that is through CBRL. Next up, we have there was a paper on spatial phenotypic mapping in lupus from Niyati Javeri. She was from the Weil Institute. It was hard to follow because I think a lot of stuff they were sharing was proprietary, but basically they were doing this complex staining of tissues with spatial immunophenotyping. So many color IHC, or maybe it was some type of in situ hybridization, was not clear. But they were regular stains, not IF stains. So like multicolor staining. They showed first using a kind of a proof of concept and other work in immunotherapy resistant and sensitive skin tissues from cancer, that they could detect metabolically active immune cells in the more resistant cells. And they went to lupus, did a neighborhood analysis, and similarly showed there that there were more increases of CD4, CD8, and proliferating cells, and they had higher metabolic activity in lupus. So that's talk 2. Talk 3 was showing about LNK SH2B3 and how it modulates activation of CD8 T cells. And for type 1 diabetes by Tarun Pant. So SH2B3 is an intracellular adapter protein for the JAK-STAT pathway. It's a negative regulator, and it works by when JAK-STAT is induced, it gets induced, and negative regulates by binding the SH2 domain back at the top of the pathway and blocking its activation. And so they show that CRISPR knockout of this have worse DM1 from loss of, so type 1 diabetes from loss of the negative regulator of JAK-STAT, and they show that this is indeed driven through STAT5 and is driven by more mitochondrial biogenesis, which increases proliferation and effector cell function in CD8 cells. Rounding out, because we eventually get to other talks on microbiome, but kind of sitting still with metabolites and such, a paper by Angela Thornton from the NIH showed there, or an abstract, shows that Helios, which is a zinc finger, is an important regulator of T cell function and Treg function in particular. If you knock it out, you actually have acquired lipodystrophy and a lack of white fat. But in exchange, instead of having white fat and adipose tissue in the mice, you instead gain a fatty liver and insulin resistance. And so this is known that adipose tissue regulates immune cell crosstalk are very extensive. What they see is that there's a T cell expansion in the adipose tissue in the knockout, less IL-33 receptor on the Tregs. The IL-10 is interestingly the same, but they get more interferon gamma producing CD8 cells infiltrating into adipose tissue and destroying it through granzyme and FASL dependent mechanisms.
Okay.
So it's killing your fat cells.
Yeah, I mean, Tregs are known to infiltrate fat tissue.
Well, this is screwing up the T cells, leads to CD8 cell infiltration and bye-bye fatty.
Yeah.
So it's your new not your new Botox, your new liposuction or CD8 cells.
Yeah, but then it goes to your liver. I'm not sure that's a good trade-off.
Yeah, well, that's in the mouse model where it's permanent, right? You just need to get rid of a little bit for a bit.
Yeah.
So then there was a paper on EEF2K, known as eukaryotic elongation factor 2 kinase by Haoyun Peng, that was showing that it regulates CD4 immunity, and knockout mice in an arthritis model have more CD4 cells with increased OXPHOS and reactive oxygen species, and it's a STAT3 mediated effect, and that T-cell transfer colitis was worse than the knockouts due to having more CD4 function. That model is known to be CD4 dependent, but a STAT3 inhibitor blockades it. And then before I pass it off back to microbiome land, we got a quick paper by Vidyaneth Chaudhry showing that plasmacytoid dendritic cells are regulated by ER-mediated stress responses. And so these are naturally interferon-producing cells and antigen-presenting cells, and he was showing that their ability to produce interferon gamma is regulated by ER stress signals.
All right. So another talk that I liked was from a study on dengue vaccination by Patrick Ilunga Mbingabo, in which they were characterizing some particular antibody responses against dengue. And they show that there's this particular antibodies that are binding a quaternary antigen that occurs when you have two of the envelope proteins that are these monomers make dimers, and then they generate a new epitope that is targeted by these antibodies. And so they show this is part of a larger study of vaccination, and they show that this is in the Philippines. So they're looking into the effects of vaccination, into protecting kids against disease. And although I think that in general, the vaccination didn't seem to help very much, they do look into the effect of having this dimer-specific antibodies that seems to correlate with better immune responses and cross-reactivity across different serotypes of the virus, and the neutralization in vitro studies. And also when they look some epidemiological analyses, it seems to also correlate with protection. Which I think is cool, because sometimes it's important to look at this. It must be a little bit more difficult to find these epitopes because they are dependent on this dimerization. But it does look that it's a good target for finding protective antibodies. I mean, we know dengue is quite a difficult disease to vaccinate against. It has been evading most of vaccination efforts so far, and this vaccine also seemed to be similar. So Patrick Mpingabo from the NID at the NIH. And so another talk that I liked was from Claire Otero from the Permer lab at Weill Cornell, and she was looking into CMV responses, and at how CMV responses seem to evade antibody protection, like they seem to evade antibody dependent mechanisms of immunity. So she kind of looked into, they had these rhesus monkeys models, and they find that, so there's two proteins from human CMV that are binding to IgGs, and they prevent FC receptor a function. And they find, so this is in the human situation, and then they find that they can find in a rhesus specific virus. So they have rhesus monkeys models. So these viruses, they can also find homologs that also prevent FC receptor binding, and they show that they can interfere with the FC gamma receptor activation, and that deleting these receptors from the CMV virus can attenuate viremia in this rhesus animal models. And so this really seems to depend on the CD4 orchestrated B cell response in a way. And this is, I guess, is very important, especially in the case of people with AIDS that are known to be susceptible to CMV infections. And basically, they see that these receptors are important in the viral evasion of FC-mediated effector immune responses. So they might be targets for therapy against CMV. And I think also the case of Mirella Cucca from the Vita Salute San Raffaele University in Milan. And she also was looking to CMV, and particularly in the balance between Th1 and T follicular helper differentiation in the case of CMV infection. Because again, when you're looking into antibody responses, you see that there's some kind of deficit in antibody responses against CMV. And she kind of shows that there is, this was in a LCMV model, so on a murine models, they show that there is a suppression. So if you have a CD4 upon LCMV infection, you have CD4 responses that are initiated, and the acquisition of a Th1 profile upon infection by CD4 cells seems to be directly inhibiting the development of T follicular helper cells. And this has a negative effect on the antibody response. And this is mediated by interferon gamma, produced mostly by the interferon gamma produced by the CD4 cells in contrast to other sources of interferon gamma. So that seemed to be the CD4 cells, and that this kind of CD4 derived interferon gamma seems to be key for the suppression of T follicular helper development. And again, so this has negative effects on antibody responses. Can we use that? Can we target that? I guess that is the future of this particular research. So very interesting talks. I really like them. What else? What else do you bring to the table today?
I bring bugs.
all them bugs in your gut.
All them bugs in the gut. All right. So we got intestinal lamina propria stromal cells. Function as a target of androgen and microbiome metabolites for the prevention of lupus by Jing Ma. So essentially, in lupus models, they noticed that women were more susceptible, or female mice were more susceptible than male mice. But if you castrated the males, they were the same as the females. Mm-hmm. And so they linked this through some work to low CD103 expression with there's low androgen levels. And then they showed it wasn't a direct effect, but it was lamina propria cells with high levels of androgen receptors. And then through microbiome transfer studies from male mice to female, or castrated male, they found it was a metabolite of the microbiome called phytanic which led to increased retinolic acid synthesis, driven by higher Bacteroides and lower Clostridium and all Stipes genuses, right?
All the
and
genuses.
All the genuses belong to us. But it was due to these microbial metabolites acting on the lamina propria cells, which then have a downstream effect on CD103 DCs.
All right. Okay.
Next up on things the microbiome does, estrogen receptors are important in EAE, from Stephanie R. Peterson. So isoflavone metabolites, so plant flavone metabolites, are a important driver in MS or regulator MS. So MS patients have gut dysbiosis with altered phytoestrogen metabolism. This group has previously shown that Equol, which is produced by bacteria, is protective in EAE. And here they've shown that estrogen alpha receptor, or receptor alpha, is upregulated in the colon of isoflavone-fed mice, and that CD8 T cells are required for isoflavone protection in EAE. Knockouts of CD8 knockouts have worse EAE. That's autoimmune encephalitis. So basically a mirroring model of MS. And there's more of this receptor on the CD8 cells. But here's the cool thing. These CD8 estrogen receptor T cells, so CD8 T cells with estrogen receptors on them, don't behave like normal CD8 T cells. They have lower levels of interferon gamma, IL-10, and other cytokines that you associate with CD8 cells, and they don't know what they're doing yet. And so there's more to be done here.
All right. Open questions?
Ooh, yeah, some open questions. People were very excited about that talk. And then human gut commensal support development of ocular autoimmunity and retina-specific T-cell receptor transgenic mice by Amy Zhang. This is near and dear to my heart. Uveitis is an autoimmune condition against the eyes. It's something that people with inflammatory bowel disease like myself are predisposed to. The blood-retinal barrier is known only to allow activated T cells in, and yet these autoreactive T cells, which have already been established, are attacking the eye. So where are the T cells getting their homing signals and becoming activated and having antigen recognition? And they show that in a spontaneous model of uveitis, that the human microbiome transferred into mouse can cause it from healthy people, and less frequently than SPF. And there's doing the T cell receptor epitope mapping now, but they show that less likely disease has more firmicutes, which are kind of the main thing that of the healthy bacteria you try to transfer in. So dysbiosis is generally associated with less firmicutes, including an IBD. So there we go. 4 bug papers, all back to back. Sorry, I'm calling them papers. That was 3 papers. But
all right.
Abstracts, presentations. The other conference I came through, they call them papers regardless. So papers they are for me right now.
Okay. Great first day. It's going to be tough tomorrow. So many talks. We're going to have to be more concise. But at least today we got to talk about all of them.
That's true. And so this is the end of our first episode of the Immunology 2023 coverage here. Don't forget to follow us on Twitter at @ImmunoPodcast to find out what we're up to at the meeting. We are taking lots of selfies. And visit us at the Immunology Podcast booth on the exhibitor floor, where you can win a gift basket. Check back here again tomorrow for another episode recapping day 2 of the meeting. See you then.